welcome Sir Michael Houghton to Defining Moments Canada, having this interview for the NobelCanadian project. As we've discussed, the idea is to create a space for Canadians to learn about Nobel laureates. So thank you for taking the time to speak with me this morning.
My pleasure.
And I've got a few questions prepared. It shouldn't take too much longer than maybe half an hour, but we'll see. It depends how verbose you are. I know I'm very...
Well, I warn you, I can be quite verbose.
Okay. All right. Well, we'll try to keep it to a reasonable timeline then.
I'll do my best.
So at first, I'd like to ask you, we're talking about commemorations, like I said, of Nobel Prizes. Can you discuss the research that earned you the Nobel Prize?
Yes, it goes back to the 1980s and 1990s. I was working in the United States at that time. I was working for a startup biotech company called Chiron Corporation. It had been started up by a couple of University of California professors, and I was sort of an international import into that company, having trained in England. And my wife and our young baby son decided to have a challenge and go to California and join the startup company. So that was really the time period and the location.
Can you remember what first got you interested in this field of study? Was there one motivating factor or something that happened that made you say, you know what, I want to study this?
Well, when I went to this company in California in 1982, I had been working on human interferon genes. So I was a molecular biologist. And at that period in history, molecular biology was going through a renaissance with cloning technologies, sequence technologies, nucleic acid sequencing technologies, and so forth. So it was really blooming. So I was an experienced molecular biologist in the human interferon gene system, and I went to Chiron thinking that I would continue that work. That was my personal intention. When I got there, I had discussions with the founders, Dr. Rutter and Dr. Penhote, and they were fairly lukewarm on continuing my work on interferons. They weren't against it, but a little bit lukewarm, I would say. And around that time, the then director of virology, Dino Dina, introduced me to the problem of non-A, non-B hepatitis. And all the methods that have been used in the last 10 years or so to identify hepatitis A virus and hepatitis B virus, they'd all been tried by groups around the world and had failed. And so Dina said to me, well, maybe, Mike, with your molecular biological experience, maybe applying molecular biology techniques to this problem will solve it. And I liked that prospect, and I agreed that it was feasible to discover the virus with molecular biology. And so I switched. And that was how it happened.
And has your career kind of stayed in that direction? You made that switch, and have you ever gone back, or has that switch been permanent?
Well, it turns out life is interesting, isn't it? How it all unwinds. But it turns out that the leading therapeutic for hepatitis C patients was type 1 interferon, which I had worked on. So it was neat. And then more recently, a new class of interferons have been discovered, the type 3 interferons. And one of my postdocs at the University of Alberta worked on type 3 interferon and came up with some nice new observations and findings. So yeah, interferon and Hep C have gone together actually very closely.
It's wonderful to see kind of how things are tied so naturally, and you would think they're not, and all of a sudden it is. So you've had a long career dealing with interferon, dealing with hepatitis C. Is there something that you would consider the most exciting piece of research or the most exciting thing that you've done?
Well, I think medically, there's no doubt that the most important contribution that I've made is the discovery of the hepatitis C virus. We knew it was in existence since 1974, 75. But until we published our papers in 1989, so 14, 15 years later, it was just known as a hepatitis problem, but we didn't know what was causing it. So I think identifying the causative virus was clearly the biggest contribution I've made to medical research. Scientifically, I would say it was that, but a couple of years previous to our 1989 papers on Hep C discovery, my lab, once again at Chiron, had discovered the genetic structure of the hepatitis D virus, which can be a very severe hepatitis around the world. And what we discovered is that it's actually very reminiscent, very similar in structure and replication to plant viruses. So scientifically, that was really a big surprise that we've got these kind of archaic plant viruses showing up in human beings and causing disease. So I think those two discoveries are the ones I guess I'm most proud of and the most interesting.
Definitely interesting, and like you say, the impact is felt and has been felt for years and years in the medical and scientific community, right? From these things that you found.
Yeah.
Curious because I'm not a scientist myself. I deal more in history, but I obviously did some research before speaking this morning. I hadn't seen what you discussed about hepatitis D. Is that something that you found by accident while you were looking for the non-A, non-B, or is that something that you also were looking for?
Great question. And the reason my lab got involved in it is that there was some evidence from the NIH around 1985, 86, that perhaps the hepatitis D virus might be related to the hepatitis C virus. So, you know, in those days, we had no molecular handle for the hepatitis C virus. You know, as a molecular biologist, you need something. You need a hybridization probe. You need a bit of nucleic acid that will bind to the Hep C genome. You need an antibody that might bind some of the viral proteins. Or you need a tissue culture system, which was the breakthrough in the HIV discovery. But we had none of those. We tried, but like everyone else, none of that worked. And so in order to try to track down Hep C, given the NIH's work suggesting a potential similarity, we actually dove in and characterized the hepatitis D viral genome, found it to be this very strange, primitive, plant-like genome. And then, having characterized it, we used it as a bait, so to speak, or a magnet. Hybridization probe is the technical word, for Hep C. But unfortunately, it wasn't related and it didn't work. So it was a detour, but it was a very fascinating detour. And medically, it helped because having identified the genome and cloned it, we were able to make a lot of recombinant protein, and we actually improved the diagnosis of hepatitis D quite a bit. But yeah, unfortunately, the reason we got involved in it was to try to capture the Hep C virus, and that didn't work because they're very different viruses. We know that now.
And that process was I think 7 years long. You mentioned from 1982 to 89 to identify what was that like for 7 years you're looking for something and you're not finding it? Is scientifically or medically there must be, is there frustrations, or is that normal to look for something for 7 years?
Well, yeah, it was a lot of pressure, to be honest. You know, I was then a young scientist and a young person, and I was in the big world of California biotech. And yes, there were lots of investors willing to put money into this new company founded by well-known University of California researchers. But big money comes with pressure, and it still does. And so, Yeah, it was a lot of pressure. You know, within the company, we had other activities from other people that were going quite well. And so, yeah, there was some finger pointing. This was one of the few programs at Chiron that was self-funded. And the reason for that is that it was so risky and so challenging that not many people would invest in it. And so Chiron was paying for it itself. So there came quite a bit of pressure, especially with repeated failure. You know, 7 years doesn't seem that long in many ways, but at the time, 7 years to try to find a virus did actually cause some pressure and some friction within the company. And I became totally dedicated to it, and so I would have continued it whether I was at Chiron or not. But eventually, it turned out well, so that's good.
Hep C became kind of your white whale, as we say. You're looking for it no matter what.
Yeah, I intuitively with my experience, I felt there must be some way to find it. It's interesting today. A lot of molecular biologists say, well, yeah, I mean, that shouldn't be a problem. You can identify Hep C in probably a couple of months now, which is true. You can with the modern technology, but back in the 80s, it was a completely different kettle of fish. And we had to be very inventive. And by the way, this was a team effort. I had my own lab at Chiron, but had a really good researcher in the lab, Dr. Chu. And also my next door neighbor, we used to talk a lot. He was working on something else, but he gave me a lot of really good input and some ideas. And eventually one of those ideas that we had discussed for quite some time actually turned out to work, but took a lot of persistence, and it took lady luck as well. I think I am a proponent. If you work hard and long enough, you make your own luck. And I kind of believe we did by not giving up and staying on to the bitter end, which turned out to be a sweet end.
Well, as they say, you have to be good to be lucky, but you have to be lucky to be good.
Aha, that's good. I like that.
I didn't come up with it. So you won your Nobel Prize in 2020, or parts of a Nobel Prize in 2020 for the work that we've been discussing that you did in the 1980s, culminating in 1989. There's 30 years in between. During that time, did you expect a call to come in? Were you expecting to maybe win it one day, or was it a surprise when the call came?
Well, I thought, given the medical significance of our discovery, that it certainly was a candidate for the Nobel Prize. And as the years went on, I became even more surprised that the Nobel Foundation had not honored the field with a Nobel. Because, we very quickly after discovering the genome of the hepatitis C virus, we were able to develop the first ever diagnostic tests for this virus. It was a huge problem in the blood banks around the world. It was causing a lot of transfusion-associated hepatitis. And approximately in the 80s and early 90s, if somebody went in and had a blood transfusion for a serious ailment, there was round about a 5% chance of getting Hep C. So it was a huge problem. We knew that it was a persistent virus, and that over time, usually many years, the patient could end up with severe hepatitis. So having discovered the virus after a 15-year hiatus since we knew some kind of virus existed, and then developing blood tests that eradicated Hep C from the blood banks, I was surprised that it took so long to be honored by the Nobel.
I like the way you say that it took so long for the field to be recognized, because as you said, it's a team effort and it's a field of so many people who contributed to it, that you felt, how come this field is not being recognized, and not how come I'm not getting the call myself.
Yeah, I never, I got into medical research to make a contribution. In fact, this morning I was writing an email to a young scientist, and I was just trying to give her some advice. And I said, I got into medical research because I really loved it and was passionate about it, having read about Louis Pasteur's life. And I got in just to try to make a contribution. I wasn't expecting it to be a big contribution. I just wanted to try to make a contribution, however small. And yeah, I mean, and that was my attitude, really. It was trying to do something useful with my life, and something I enjoyed. But of course, once we discovered it, it was big news. And then once we cleaned up the blood banks around the world, very quickly, within a few years, we stopped hepatitis C transmission. I mean, in the United States alone, I think at one point it was causing like 150 infections every day from the blood banks of the country. Just to give you a sort of a rough idea of how important this was. And so I think once we had done that, I thought, well, we've got to get a Nobel for this. But it wasn't, I've never been worried that much or cared much about awards. The only time I do get some consternation is when every year when the Nobels are announced and we're overlooked. I thought, well, this is strange. I mean, it's a very important virus, and we solved it. I don't understand why it's not being honored, but eventually it was. So everything turned out well.
And there's always concerns, and it depends who nominates and things. In doing this project, we've been looking at kind of the process as to, when people are nominated, how it works. And there seems to be, if there's a rhyme or reason, we haven't found it yet. It's kind of almost a randomness to why certain years, certain things are recognized and why not.
Yes. I'm a strong advocate of reform in major prizes. I'm in some ways, I feel I shouldn't be advising the Nobel Foundation and how to give their Nobel Prizes. They've done quite well on their own for the last 100 years. But I do think in this age of multidisciplinary teams, international teams, and also in order to motivate our students correctly and to teach them how you should behave during your career, I think it would help if major awards like the Nobel, like the International Gairdner Award in Canada, like the Clinical Lasker Award from the USA, those Gairdners and Lasker Awards are considered the baby Nobels. I really would like to see them be more inclusive, because, the basic truth is that the hepatitis C virus was discovered by the 3 people that were honored by the Nobel Foundation, and it was a huge honor, of course. But there were other key contributors, and I would say there's probably another 5 or 6 other scientists that deserve to be honored. And what is the harm in being more inclusive? If it's a team of, sometimes, in the case of Albert Einstein, right, he was a genius and he can stand on his own. People say he deserved to win 4 Nobels just on his own. But often, more usually, it's large teams. And why not honor the larger team rather than a subset, albeit leaders, but they weren't themselves just responsible for the success.
And it seems, in speaking to Nobel laureates and doing this project, it seems that that's a shared sentiment. We spoke to Art McDonald, who won a Nobel Prize for physics, and he won't say I won a Nobel Prize for physics. He'll say I was awarded on behalf of the Submarine Neutrino Observatory collaboration. I was awarded a Nobel Prize because I was the leader, but there's 700 people who worked on this for 40 years.
Well, I commend him for that philosophy, and that is mine. I really would have liked a larger collection to have been honored by the Nobel, but that isn't that isn't their history. You know, they have a maximum of 3 people to be honored. I really would like to see it change, because who loses? It seems to me there's something to be gained, and also a great message to go to our young students and trainees, that you have to work together. And when you do, make sure that, or as far as you can, make sure that all the key contributors will be acknowledged. Of course, you know, there'll always be some frustration. Even if you made it the rule, there could be 10 Nobels for a particular subject. There'll always be people that are disappointed, but at least you make sure that the inner electron orbits, so to speak, are included.
That seems to make perfect sense to me when you explain it that way, and I'd be curious to see kind of it might just be conservatism within the organization, as this is how it's always been, we'll keep it, because
Yes.
the Nobel Prize for Peace can be awarded to collectives and to organizations.
Mm-hmm.
It's the only
Yes,
one.
good point. Yes,
So
good point. The only
why
one.
not the others?
Yeah. I'm not criticizing the Nobel Foundation. Kudos to them. We all know, and I've learnt in the last 2 years, the tremendous resonance of the Nobel Prize. Everyone associates it with brilliance and contributions to the human species. And so it's been very successful. But I think they can still get that success by being more inclusive, and so can the Lasker Award, and so can the Gairdner Award. We've got to work together more and more, right, internationally in all fields, and this could be a good way to do it. I often talked about, our late Queen Elizabeth II. She instituted a new award in the UK after her name. And originally, the rules were like the Nobel and the Lasker and the Gairdner. No more than 3 people can get it. But straight away, they modified it. And I think the first award that got the Queen Elizabeth II Award for innovation and science was a team of 5 or 6. So traditions can change.
They can, yeah. Difficultly, but they can.
Yeah.
You mentioned that in the last few years, you've noticed really the impact of the Nobel Prize, kind of, I guess, the weight that it carries. Can you speak to that a bit? It's not in my prepared questions, but I'm curious to see how your life has changed, or how your career has changed since you've been awarded the award.
My life has changed by becoming much more busy. So it feels like everyone wants to talk to me. Everyone wants me to give seminars. And of course, Nobel laureates have a duty to respond and do that. Much of the time you're talking to student groups or trainee groups, and you're trying to help them have successful careers and help them along their pathways. But you do get incredibly busy. And I think for me, it's kind of, before and after the Nobel, I haven't changed. I'm still the scientist I was. But, people, especially maybe students and the general public, you get a Nobel, they think you're some kind of super being, the cartoon movies of X-Men, is it?
Yeah.
They suddenly feel that you're a super being, and well, I'm just the guy I was 2 years ago. I'm trained and qualified and worked hard with a lot of good colleagues, and we succeeded in a nice medical challenge. But I've been a bit surprised just how people view you. They almost view you like you're some kind of god. I remember one of the first team meetings I was at after the announcement, and I was on Zoom because of COVID, and I remember one of my colleagues on that large team said, "Wow, I don't know I don't know how to respond to you. I'm seeing you as a Nobel laureate. I feel like I don't belong, you know. It's like, but wait a minute, we've been talking for years. There's no difference. So it is amazing to see just the resonance. I'm not sure if that's the right word, but how the general public and even scientists view the Nobel laureates. They're quite amazing.
Yeah, I probably won't win a Nobel Prize in my life. I don't know that I'm on the way towards it, but from everyone I talk to, it seems to be so kind of unfathomable how people will treat you from one day to the other. It's hard to imagine that change.
Yeah. Well, I think probably it is due to laureates like Albert Einstein, you know, and other physicists, Niels Bohr, and other great physicists really set the tone of the Nobel Awards. You know, these truly were brilliant geniuses, you know. And, you know, I'm still just amazed about Einstein's mind, you know. Some of the things he predicted 100 years ago were only now just getting experimental proof of. And, you know, what a mind. And I think really I think maybe it was those physicists in the early days of the Nobel system that really impacted the general public and associated with the Nobel with brilliance and genius. I don't consider myself a genius. I consider myself well-trained, willing to work with other people and to persist, and that's fine. But I think maybe that's why the Nobel has such incredible effect today.
I'm sure the reputation of first laureates, of famous laureates, kind of brings that gravitas to it. But I also am curious to see if at the time the laureates who are now today kind of legend felt kind of similarly, that, oh, I'm well trained, I have a good education, I'm not necessarily a genius. And then, you know, 50 years later, now we look at them and go, oh my God, this person won a Nobel. I'm not like that, but this person was. Maybe in 100 years,
Yeah.
you know, a certain
Yeah.
lore will build around yourself or other contemporary winners.
Yeah, I mean, I think obviously to win a Nobel, have got to have done something very important scientifically at a basic science level or at a sort of translational level in medicine or physiology, etc., or economics or some other area. You know, so I think what we did with Hep C, you know, was a major event. The world had been trying to find it for 15 years and had failed. And each day, you know, probably around the world, you've got thousands of people being infected from the blood supply alone. So I'm not diminishing the impact of what we did to discover hepatitis C virus. You know, it was a substantial contribution to medical research, and it needed a lot of experts working on it, and it needed persistence. You know, persistence is really important in work that I do. But I think there's a public reflex to think every Nobel laureate is an Einstein, you know, comes up with these theories that are just so basic to our existence and will last 100 years. Well, I think we will be in the history books in 100 years' time. And, you know, modesty aside, I think we probably deserve that. But on the other hand, that's what we're trained to do. That's what we're paid to do by the taxpayer. And that's what we have to do. So anyway, I'll stop there.
But you're absolutely right. You're doing what you're meant to be doing, and you're being recognized for it. And it's, you know, it doesn't mean that the recognition is not legitimate just because you're doing what you're meant to be doing. You're doing what you're meant to be doing excellently, I guess, is the idea. So one question that has to do with Nobel, because we're doing a commemoration on Canadian Nobel laureates. And so far we've talked about how you're from the UK and you did work at Chiron Corporation in the US. There's got to be a Canadian element in there. So
Yes, indeed.
you won while you won your Nobel Prize in 2020, you're at the University of Alberta.
Yeah.
Can you talk kind of how you got to the University of Alberta and why you made the transition from kind of corporate research at Chiron to academic research at the University of Alberta?
Yes, I so being a member of the viral hepatitis international community, I became aware of the work of Dr. Lorne Tyrrell, who's a very well-known researcher and clinician at the University of Alberta. And I admired the work he and colleagues had done in the field of viral hepatitis. I got to know Lorne a little bit, and I liked him personally. And having spent many, many years of my career in the private sector, I thought, well, it might be nice to go back to academia and spend some of my career in academia. So I was in San Francisco on a sunny morning, driving to a company, an antiviral company. It was a startup. This is long after my work on Hep C. And I had joined as chief scientific officer of this antiviral company. And I got a call from Lorne Tyrrell, and he said, Mike, there's a new program in Canada being funded by the federal government to try and attract international scientists to Canadian universities. And these are big grants, and they're 7-year grants. Do you know anyone who would be interested? And Lorne said that it was a loaded question deliberately. He was hoping I would say, yes, I know someone, me. And that's what I did say. So, I said, yeah, sure, I would be up for it, Lorne. But, my family's based in the San Francisco area. So I could only do it if I was part-time on site. And he said, well, the university would be willing to do that. And so that's how it all happened. And I came to the University of Alberta on a Canada Excellence in Research Chair program from the federal government. And I must say, I really, really enjoyed it. I've been now at the University of Alberta for 12 years. It's been interrupted quite a bit by the COVID pandemic in terms of traveling back and forth being difficult. But being at the U of A, I've really enjoyed. It's, in a company, you have a lot of very good scientists, but on a huge university like Alberta, there's so many really good professors and labs around, and you interact with them. So, for example, I've met some really wonderful scientists in computational science, and they've been pioneering computational drug discovery. And so I've latched onto that big time. And, you also meet some outstanding young scientists, that have real intellect and energy and training. So I really enjoyed my time at University of Alberta. And, by the way, I've continued my work on developing a Hep C vaccine. We still don't have one after all these years. I worked on it when I was in the States, and I've continued to work on it at the University of Alberta. And, once again, involving a lot of other people in the lab, really good young people, we've made some further inroads. And so, I think that was a factor in getting the Nobel, that with a new team, I was still working on Hep C and trying to prevent even more infectious events in other ways besides the blood supply.
Yeah, the award could have been, it is an award for your work in the 80s, but also continued your career impact on the field.
Yes, I think so. We had at the U of A, we published several papers, which were, I believe, seminal. So a young research assistant, Dr. John Law, is in my lab. He showed that in humans that were vaccinated with my prototype Hep C vaccine, that they were able to make neutralizing antibodies, but they were broadly cross-neutralizing, because Hep C is like COVID. It makes a lot of variants. And so the big issue is the vaccine, is it effective against all the variants? And John actually showed for the first time in humans that the vaccine could indeed elicit broad cross-neutralizing antibodies, which is exactly what we want for the COVID vaccine. And to a large extent, we do have, albeit you have to keep coming up with new vaccines against the variants. So, that was a seminal paper. And then a student in the lab, Jason Wong, he published a paper in 2014 where it explained why we get broad cross-neutralizing antibodies. And he showed it's because there are multiple antibody combining sites on our vaccine, which is responsible for the broad cross-neutralization. It's not just one. There are many sites on this complex virus surface to which antibodies bind and individually can neutralize different strains. You put it all together, and despite it being a very heterogeneous virus, a single vaccine is surprisingly effective. So my work on Hep C did continue, and it still is very much so at U of A. So I think that was a contributing factor to the Nobel Award.
If it wasn't, it should have been, because it sounds like the work is still extremely meaningful, and you're still coming up with things that are adding to the work from the 1980s, right? So
yes, WHO inform us that there's still around 400,000 people dying of hepatitis C around the world every year. Unfortunately, with the opioid epidemic, we're seeing an increase in incidence in many countries, including Canada, rather than a decrease. So how do we stop that? Well, we need a vaccine. So I'm hopeful that by the end of this decade, we'll have a Hep C vaccine. And then that will be the final nail in the coffin of Hep C causing disease.
Well, hope that you continue that work and get to it. I won't take too much more of your time, because I'll let you get to that work. It sounds like it's urgent. But so I want to thank you for taking the time to speak with me this morning. The idea is for students and teachers to get to watch this video and other resources for Canadian Nobel laureates, and to learn different routes to the Nobel Prize, and also what the Nobel Prize, what it highlights, what it awards, what it celebrates, and showing all these different paths of Canadian excellence. So thank you for taking the time, and I hope that this will become a resource that will be really well used by teachers and students alike.
Thank you very much for the opportunity, and thank you for what you do, because communication and education is so important, obviously, but thank you for what you do. I appreciate it.